There we go. There you are.
Yeah, I think argument is more defensive. So we're like, well, I mean, it's gonna happen.
Yes. I messed up because I was testing out the stream like when we were getting it ready. So I tested it and then I clicked done.
Oh, then
it was like, okay, you're all done. And it's like, oh, there's just this video of me sitting in front of the computer making like it's on her live stream. So I had to like go through and update all the Facebook. Like hit everything like
How do you been?
I'm good.
Yeah, I got it. Yeah. Got it approved online yesterday.
Oh, that's awesome.
Electronic approval. I got his signature for it. That's good.
Okay,
So, then I handed it. Yeah, no, the registrar was a lot of that.
But it's already
one day turn. Yep. Yeah.
What is that about your research? What's the
Yeah. Usually, exactly. Translate the antibodies.
Okay, cool. That's really cool.
Yeah.
I guess, have you raised money for it?
In the process, we're in the process. That's one of the things, touring lab facilities, plus the incubators and other kind of spaces like that in the area. And then the farm all the way up.
You like watching
I know.
Yeah.
I mean, that's cool.
It'll just be like day one. But we already have like a template ready for so basically once we can finalize the funding, hopefully that's set stage
So what's funny,
but yeah, it's been a lot of fun. We have an interim CEO that I do, or I've been learning from along the way. All the legal stuff.
Yeah. Oh my gosh.
Yeah, equity, corporation, council,
Yeah.
Other vocabulary. Yeah, that's Oh yeah. It's fun. It's variety. These things are just generally easy to know about. I think like good to know how to work.
Are you going to take over?
No. What we're trying to do is hire someone. Because there's so much regulatory and medical experience you need to have in doing that. It's a lot different. If you're just like a subject matter expert, you kind of be a CEO. You're like, you gotta have a network of people. A lot of probably end up staying closer to the sun.
Well, you'll be able to do whatever you want.
Yeah, early, I guess everybody's busy.
Yeah, it doesn't matter really what it says.
I'm also taking up the trash.
Yeah.
I think I recommend I'd have to get you now. I'm not saying
So, fall running.
Yeah, you do not. I decided.
originally my plan was to
Yeah, yeah. Yeah, that was a big issue.
What
So like Obamacare, just the outskirts. Obamacare ends. Well, the calendar year starting September 1st, so it ends October 31st. I'm on writing course in summer.
You had another.
No, no, it ends on the 31st.
You said October 30th.
Sorry, August.
So yeah, then we're getting
August 30th. Yeah, exactly. You can't read wrong. This year now.
How did you know?
So you basically have to just on the cover California, like the Obamacare. Figure it out.
Yeah, that was another good thing. We didn't even realize until after I made that decision because that she was going to go on.
Yeah. Yeah.
When are you up to that?
Yeah.
Oh yeah.
Yeah. The individual life expected. But a calmer runway exit.
Yeah, it definitely does because now I have for
Yeah. Well, like things could be now on your schedule, right?
Yeah.
It's not like we have to move out 3 days and find a place.
Yeah. That's kind of what because that's what it's gonna end up being. It's like, well, we're gonna be moving out here. Better the fence like a week later.
Everything at once.
Yeah.
Cool. You've been doing kind of business related stuff, right?
Yeah.
That's the area you want to stay at?
Yeah, so I keep going into it. I'm going to be doing, you know the groups in my
Mm-hmm.
So we're doing this so just helping work with partners that they whatever those partners their message out. So I started doing it part time. There's no way to manage it. Yeah, I mean.
Oh, set up.
Yeah. Glad to have you here. You're gonna be really
Yeah. Can you reveal that went you know, in front of everybody, they actually
Alright. What was the best step? I'm still thinking with it.
Yeah, we're trying to be a lot of people learn about
Apparently, like,
I'm good, thanks. Yeah, Murderers Row. Do you know the game theory of the
I'm excited to hear about it.
What's the success rate? Of course. How long are you doing? See you. From other countries, you forget what
Yeah. I remember getting
I have to be careful. Yeah. Drop those steps like that. Somebody would come and be like, oh, I really get it. More than 50%.
I'm just wearing it now. He'd be like, yeah. Okay, everyone for the last week has been like, Stephanie, you need to focus. Like, I know. I
Do you want some food or something?
Yeah. Thanks for having me.
Yeah, of
She really wants to go to Yale, but number one. 0% of things. I read it today.
I love the God, now I hate my
Hey, how are
Okay.
And there's a park. And there's a park. That's beautiful. Let's go. I would like, hey, have a look at it.
Oh, that's so this is the folder you go to school. So make sure it's in the right box. Yeah, I'm just like literally. Yeah, so literally. And we're just waiting on. Right? So apparently, you know, it's fine. Okay, good. No fine.
Oh, yeah.
Okay.
All right, why don't we get started? My name is Steve Quake, and I'm going to introduce Stephanie Solanas today. This is a little bit embarrassing to do as an introduction because my position to the press for the last 9 months has been I don't work on CRISPR. And this gives a little bit of a lie to that, but as you'll see, it has nothing to do with what the press has been concerned about recently. Stephanie done a really lovely thesis. I'm not going to sort of foreshadow it any further, and you'll see really other creative things she's done with Cas9 and its variants, homologs, and some neat applications in lineage tracing. But I'll just say a few words about her. She's certainly one of the most enthusiastic graduate students I've had, because I very vividly remember she did not want to wait for the school year to start to begin her rotation in my lab, and was very passionate about starting in the summer. And I said, well, I'm getting sabbatical this summer. I'm not around. I want to start anyways. I want to get a head start on the whole thing. We worked it out in the end. And she came and stayed, and it's been a real pleasure to advise. It's the thesis has been a bit of departure from the things we worked on in the lab, but it's work that I'm very proud of. And we had some happy news yesterday where another paper accepted first thing in the morning. That was very timely. I'll leave it at that and let you take over, and hope you'll enjoy what she's been working on for the past few years.
Thank you for the introduction, Steve, and thank you everyone for being here today. I'm excited to have the opportunity to tell you about my thesis on the topic of using genome editing to build new tools for genomics. Our time together today will take us from the hot springs of Yellowstone all the way to the digestive tract of a millimeter-long ground. That's a lot of ground to cover, so to get us oriented, I put together a little roadmap. So I'll start off by telling you a little bit about myself, and we'll highlight why I'm passionate about building technology for biology. We'll then spend most of our time together today on two projects. The first is about using genome editing to record cellular histories, and the second is about an exciting new Cas9 protein. I'll wrap up my talk by sharing future directions and what I'm excited about working on next. I'll then open the floor for questions from the audience, and after that conclude with some personal acknowledgements. So with that in mind, let's get started. I thought you might find it interesting that I happen to come from a place that's also called the Bay Area, which is what the Gulf Coast suburbs of Houston also happen to be called. Growing up, I dreamed of exploring the frontiers of space as an astronaut. And this was perhaps not so surprising, given that NASA's Johnson Space Center was practically in my backyard, which is what I show you actually in the photo in the top right. In it, you can see the space shuttle taking a piggyback ride over NASA's Mission Control and Johnson Space Center, but you can also see that it's right down the street from my middle school, right there at the top of the photo. But at any rate, my talk today isn't about the vast reaches of space, but rather it's about the fascinating worlds that exist within even the tiniest of cells. What changed my perspective and my aspirations was the opportunity to conduct research in college in the area of tissue engineering, and I was hooked. I found that cells are self-contained, beautifully designed entities that we have so much to learn about and to learn from. From my work on bone regeneration, I was fascinated by how cells from the same progenitor could differentiate and become dramatically different, even unrecognizable. And this was a phenomenon that motivated me to pursue a PhD and come to the Bay Area, and specifically to Stanford, to delve into these concepts in a holistic and quantitative manner in the laboratory of Stephen Blake. My work focuses on creating new experimental capabilities by leveraging genome editing, and I see my work fitting into the broader field of bioengineering and genomics by enabling us to better engineer living systems, to learn from nature, and ultimately to tackle complex challenges. These endeavors are in no way disparate, but rather they are interconnected and self-reinforcing. I'm excited about the field of genomics since it's one of the best ways that we can interact with and in the process learn about and learn from the world around us. The first project I'll tell you about today highlights what we can achieve by engineering living systems. In this project, I used CRISPR-Cas9 to create updatable sequence barcodes and plus to write a cell's lineage into its own genome. This work was published in ACS Synthetic Biology, and in my talk today, I'll tell you about why we were interested in recording lineage information in the first place, our experimental proof of concept I performed, as well as the simulation and modeling work I did to determine how forthcoming versions of this technique can be further improved. So given my fascination with cellular differentiation and decision-making that I shared with you in the introduction, I became very interested in lineage tracing. Lineage tracing, or the determination and mapping of progeny arising from single cells, is a foundational technique for studying biology. Lineage tracing allows us to understand how cells are related and how they give rise to complex behaviors in development, disease, and regeneration. Lineage tracing has certainly progressed from staging and sectioning embryos, as biologists did in the 1800s, to more recently marking lineages of interest using fluorescent proteins, to using static genetic barcodes, and even using more recently genetic recombination. These approaches have enabled important advances, but we suggest that they can even be improved with respect to certain features, such as their information content, their ability to be combined with other system readouts, and in some cases, their need for real-time observation. To this end, I thought that a dynamic and updatable means of recording lineage information could be an improvement, and that a promising way forward would be to leverage the great potential for data storage of DNA. I created a lineage tracing strategy based upon the introduction of specific mutations into cells and the propagation of those mutations to daughter cells at each cell division. Towards this end, I harnessed the specific genome editing capabilities of CRISPR-Cas9 to target particular sites within a cell's genome. Before I proceed any further, I would of course like to acknowledge that this is an active area of research as evidenced by papers from several labs that I've listed at the bottom of the slide. We developed and pursued this idea of CRISPR-based updatable barcodes independently, and in my presentation today, I'll share our take on this promising new technique. So our method works as follows. The activity of Cas9 at particular sites within the cell's genome, which I refer to as cut sites, that are specified by the sequence of a guide RNA molecule, lead to the creation of double-stranded breaks that which, when repaired by non-homologous end joining, result in an insertion-deletion mutation at the cut site, and thus the unavailability of the cut site for future targeting. As an example, let's consider the following model case, in which a genomic region has 3 different specified cut sites, as indicated by the color shading in the fragment that I show at the top of the slide. Upon cutting by Cas9 and non-homologous end joining repair, an indel is created. This is what I show by the change from solid fill to dashed fill and the shortening of that cut site. This indel is transmitted to all progeny of the cell in which the indel is created, resulting in a heritable record of their lineage. In a similar manner, an indel can be introduced in a daughter cell, as shown by the change in the purple site in the bottom row of fragments, thus denoting a sublineage. It's also important to consider the case in which non-homologous end joining occurs with what I have termed dropouts. So in the event that 2 nearby sites happen to be targeted, non-homologous end joining can repair the double-stranded breaks so that the sequence between them is lost in the process of creating an indel. The removal of the intervening sequence is particularly problematic because information, such as indels that were previously encoded in the sequence, are lost. This is something we'll discuss in greater detail a little later in this portion of the talk, but first I show that even with dropouts, this strategy can capture important lineage information. As a proof of concept, I turn to C. elegans, the millimeter-long nematode that I mentioned to you at the beginning of my talk. It served as an important model organism. It's well characterized, capable of self-reproduction, and with a quick developmental timeline. These worms are known to have a fixed number of cells. In the late 1970s and early 1980s, the complete lineage of C. elegans from a single-celled egg all the way to an adult was determined. Now, this Nobel Prize-winning feat was accomplished by painstaking direct observation of the organism under the microscope as it developed. This was possible because C. elegans is clear, and it remains the only organism for which a complete high-resolution lineage tree is available. With this in mind, I implemented our lineage tracing strategy in C. elegans. To do so, I used C. elegans from the EG6173 strain, which have been engineered to natively express green fluorescent protein, EGFP, and thus they appear bright green as shown at left. Since EGFP is not essential to worm development or well-being as far as we know, I decided to use the gene encoding it as a sequence barcode. I designed guide RNA for 10 different sites within the EGFP gene, thus setting up a barcode with 10 cut sites that could be read out within 2x300 paired-end sequencing. A mixture of the guide RNAs were complexed with Cas9 to create ribonucleoprotein complexes, which were then injected into the gonide of EG6173 worms. The ribonucleoprotein complexes were encapsulated within the forming eggs and then stochastically targeted cut sites over the course of the worm's development. Now, since I had elected to target EGFP, I was able to screen for worms which had undergone effective CRISPR-Cas9 cutting because they would no longer appear green, which is what I show at right. Progeny displaying this desired phenotype were dissected to remove the intestine, which is known to constitute a distinct lineage, and then each body and intestine was lysed and sequenced separately. The EGFP region containing the sites targeted was inspected for the presence or absence of indel mutations in order to infer lineage relationships from cells from the sequence barcodes. I grouped barcodes by shared indels and used the frequency of indels observed across unique barcodes to infer the order of introduction of the indels and thus to construct lineage trees, a subset of which I show on this slide. For reference, each column is a barcode and each row is an observed indel. I found that barcodes from the intestine very encouragingly displayed an assortment of indels like the one highlighted, not observed in the rest of the body, suggesting a distinct cell type in agreement with the known lineage, since for C. elegans experts in the audience, indels created after the formation of the E blastomere should be restricted to appearing in either the intestine or the body. Moreover, in the sample derived from the intestine and that from the rest of the body, sets of barcodes contained shared indels, like the one highlighted, indicating descent from a common progenitor cell, such as the P1 or the EMS blastomere that occur prior to the formation of the E blastomere. One last thing I'll point out is the presence of barcodes, such as the leftmost two shown, that contain no indels or just one indel. This shows that the rate of CRISPR-Cas9 activity needs to be further tuned to record cell divisions over the entirety of development. Having used a more traditional phylogenetic approach to examine our data, I next wanted to quantitatively determine how well our barcoding method was capturing lineage information. To do so, I assessed its performance in correctly capturing the separation of the intestinal lineage from the rest of the body for the full collection of barcodes from the worm I showed you on the previous slide. Some of those barcodes did discuss the dropouts I mentioned to you at the beginning of my talk. Using the k-nearest neighbors algorithm, I compared the predicted tissue of origin to the actual tissue of origin for each barcode. I then calculated two instructive metrics of information retrieval. The first, recall, is the ratio of correctly identified intestinal barcodes to the total number of actual intestinal barcodes. The second, precision, is the ratio of correctly identified intestinal barcodes to the total number of putatively identified intestinal barcodes. The results were compared to those generated by randomizing the dataset's barcode assignments over 100 trials. The experimental results show that the precision of the actual data is 0.86 compared to 0.32 for the randomized case, while the recall is 0.4 compared to 0.03 for randomization. These results underscore the ability of updatable sequence barcodes to capture lineage relationships using approaches independent of tree construction. It also suggests, though, that the performance of updatable CRISPR barcodes can be further improved. If updatable CRISPR barcoding were improved, namely by addressing the sequence loss caused by barcodes, R and other approaches could be even more informative. To predict the potential of such an improved approach, I examined the diversity of indels generated using CRISPR-Cas9 and calculated the information content of the described barcoding method. I compared the indels observed across the 10 targeted sites in 8 sequenced C. elegans samples for cases in which dropouts did not occur. The distribution of positions contained within indels relative to the position of the CRISPR-Cas9 introduced double-stranded break can be described using Gaussian curve, which is what I show on the slide. This enabled us to calculate the Shannon entropy, a metric of the information capacity of the system, which I found to be 4.42 bits. The theoretical upper bound of information provided by CRISPR-Cas9 barcoding was determined using a model in which the probability of position occupancy by an indel conditional upon the indel's length was used. Making use of the parameters describing the distribution of experimental indel lengths and assuming a uniform distribution for the utilization of positions, we found that the Shannon entropy of an individual cut site within such a sequence barcode could encode 6.86 bits of information at most. Now, to provide a more realistic estimate, we weighted the occupancy of particular positions by the likelihood of their inclusion in the indel, giving a Shannon entropy of 6.61 bits. The difference between the theoretical values obtained at right compared to the experimental result can be explained in part by the bias observed in the creation of indels. After all, by describing the location of the double-stranded break with respect to the center of the subsequent indel normalized to indel length, it is evident that indels resulting from CRISPR-Cas9 activity tend to lie to one side of the specified double-stranded break, which reduces the number of possible states and amount of information able to be encoded through barcoding. This point can be further illustrated by using a modified version of the aforementioned model. By using a Heaviside step function instead to describe the occupancy of positions around the double-stranded break to determine a lower bound of performance, I calculated a Shannon entropy of 3.8 bits for a single cut site, which is still a considerable improvement over the effectively binary information that's supplied through current recombination and color-based lineage tracing techniques. For comparison, they offer only 1 bit of information for a given site or color. With 6.61 bits as we predicted in each site, and just 10 sites as we used in our experiments, one could create approximately 10 to the 19th unique barcodes, which is actually more than the number of cells in the human body. Future work is needed to develop control of CRISPR-Cas9 activity to achieve such performance. As I wrap up this portion of the talk, I'll share results of the simulation I performed to drive this point home. I simulated sequence barcodes and the resulting lineage trees in C. elegans. I represented CRISPR-Cas9 activity as a Poisson process and followed the known cell division pattern from C. elegans for the initial divisions in which major lineages are specified. Barcodes were generated to represent 2 cases in which non-homologous end joining did and did not result in dropouts, again, the removal of sequence between targeted sites. For both cases and for a range of expected values of the Poisson process, 100 sets of simulated barcodes were generated and the corresponding lineage trees constructed. Modulating the expected value of the Poisson process enabled me to explore the effects of CRISPR-Cas9 cutting rate, which I defined as the number of indels introduced per time. To compare the agreement of each simulated tree with the known lineage tree, we calculated the cophonetic correlation between the two. As cophonetic distance describes the intergroup similarity of the cluster in which a pair of observations are placed in a given tree structure, the correlation between resulting cophonetic distance matrices of full tree structures provides a suitable metric for the evaluation of concordance between 2 trees. I found that cophonetic correlation between each simulated tree and the known tree understandably improved with increasing number of indels introduced during each generation, but even with approximately 1 indel introduced per generation, corresponding to a Poisson expected value of 0.05, the lineages constructed agreed well with the reference lineage. As expected, the presence of dropouts reduced the agreement with the known tree, but despite this, for lower Poisson expected values, the difference in performance between barcode sets with and without dropouts was not statistically significant. However, it is important to note that even with the cutting rate approaching 8 cuts per cell division, the best correlations achieved were 0.99 without dropout and only 0.75 with dropouts. This suggests that in any practical situation, the stochastic generation of lineage barcodes cannot enable perfect lineage reconstruction unless the scheme is modified to eliminate dropouts. With that, I'll wrap up this portion of the talk here. In summary, I was motivated by the great potential for data storage and manipulation within DNA, and thus developed our dynamic sequence-based barcoding technique. My work demonstrates the potential of such nuclease-based barcoding methods by illustrating the agreement between the known lineage of C. elegans and that obtained using sequence barcodes. It delves into the challenge presented by sequence dropouts through a simulation-based approach, and underscores the technique's promise by establishing its information content. With this in mind, controllably introducing mutations in a desired genomic region and at rates higher than background offers the ability to track parent-daughter relationships with even greater clarity and without the need for whole genome amplification. Taken together, I present proof of concept for the tracking of cells by means of dynamic sequence-based barcodes, which I am confident will enable unprecedented and more complete studies of cell lineage. Shifting gears away from engineering living systems using CRISPR, I'll tell you about another project of mine in which I found a novel Cas9 protein in environmental sequencing data, which is very much in line with the next area of my talk, learning from nature. We discovered a new Cas9 protein with properties that lend themselves to interesting tools for genomics. I'll tell you about how I combined bioinformatics and experiments to understand and characterize this new Cas9 protein, and how I think it will be useful to a broad array of researchers. We filed a provisional patent for this work, and our manuscript just got accepted, so I apologize for these slightly out-of-date slides. Since this is a more fundamental CRISPR-Cas9 project, I thought it would be appropriate to take a step back and look at the bigger picture of CRISPR systems, which are actually the heritable and adaptive immune system of prokaryotes. It's so important to keep in mind that there's a considerable variety of effector proteins in the CRISPR genome editing toolbox, with functions ranging from integrating genetic material into the spacer array, as I showed here at the top, so that the prokaryote can defend itself from infection, to processing the RNA, as shown here in the middle, that's ultimately used to bind and modify the target nucleic acid, as shown at the bottom of the slide. Class 2 systems, shown at bottom right, and in particular, type 2 systems that involve a Cas9 protein, have been broadly implemented because these systems rely on Cas9, a single effector protein. Of course, the Cas9 from Streptococcus pyogenes, or SpyCas9, is widely used. In fact, it was the Cas9 protein that I used in the previous portion of my talk. So you might very well ask, Stephanie, why would I even want a new Cas9? We can do lots of things with the old one. So in certain cases, I suggest that it could be beneficial if there were alternatives to address aspects such as the limited delivery capabilities of SpyCas9 due to its large size, which is actually right up against the payload of an adeno-associated virus. This aspect constrains in vivo applications, so a somewhat smaller Cas9 protein would allow for greater flexibility in such experiments. Moreover, with respect to in vivo applications, important work from Matt Porteus's lab here at Stanford has revealed that most people have pre-existing adaptive immune responses to SpyCas9. This certainly puts limits on the efficiency of potential gene therapies using SpyCas9, whose bacterial host actually is one of the most common pathogens that humans encounter. Thus, a Cas9 from a unique environment could serve as a useful alternative. More broadly, not all desired targets are even available for editing by SpyCas9, since the target must be followed by SpyCas9's protospacer adjacent motif, or PAM sequence. However, PAM sequences actually vary by Cas9 homolog, so additional Cas9s can enable the targeting of previously unavailable sequences. Finally, I include a bit of an unusual item on this list of traits. SpyCas9 works at only up to 44 degrees Celsius, which is perfectly suitable for many applications. However, considering that a lot of molecular biology and genomics workflows involve temperatures well in excess of 44 degrees Celsius, it would open up new possibilities to be able to use genome editing at even higher temperatures to minimize the presence of unwanted sequences. With this in mind, new options are of interest. That being said, new possibilities do exist across the bacterial and archaeal kingdoms, and the diversity of nature broadly motivates our work in the area of mini metagenomic sequencing. Paul Blaney, who did his postdoc in the lab and is now an associate professor at MIT, collected a sample from the lower Geyser Basin in Yellowstone National Park. And Brian Yu, who did his PhD in the lab and is now a scientist at the Chan Zuckerberg Biohub, developed the mini metagenomic sequencing method and sequenced this sample. In this data, we were excited to see the sequence of a full CRISPR locus. It actually came from an incomplete genome from the Ignobacteriae phylum and contained a Cas9, which I accordingly named Ignobicas9. I was excited because it was smaller than the Cas9 protein from S. pyogenes. Given that the hot spring it came from has recorded temperatures above 90 degrees Celsius, I thought it would likely be highly thermostable. And I was motivated to see if I could get Ignobicas9 to work. The ultimate question being, at how high of temperatures would it be active? I was starting from just a sequence fragment containing the CRISPR locus, and I needed to identify and optimize 3 key components to get a working Cas9 system. First, of course, I needed to express and purify Ignobicas9 itself, which I did after codon optimizing its sequence for E. coli expression. To direct the Cas9 to the target sequence, I needed to infer the sequence of 2 important RNA components from the CRISPR locus itself. The CRISPR RNA, shown in green, contains sequence complementary to the target DNA and is complex with the tracer RNA, the transactivating CRRNA. For ease of use, I plan to link the CRRNA and the tracer RNA so that they formed a single molecule, the guide RNA, constituting the second component that I needed. Finally, to target the desired sequence of DNA, I needed to know the protospacer adjacent motif, or the PAM, which is the sequence that must flank the target. So with this in mind, I used a combination of bioinformatics and experiments to learn more about each of the 3 components, the Cas9 itself, the guide RNA, and the PAM. Starting with the Cas9, I was interested in understanding how it might behave. Aligning its sequence to that of SpyCas9 allowed me to identify important functional domains and to verify that it contained the HNase nuclease domain, which cleaves the complementary strand, as well as the RubC-like domain, which cleaves the non-complementary strand. Thus, we could expect double-stranded breaks from this protein. You'll probably notice that Ignobicas9 is shorter than SpyCas9, which largely comes from the shortened recognition lobe shown here. This is consistent with other smaller Cas9 proteins. At right, I include a homology model of Ignobicas9's structure based on the crystal structures of other characterized Cas9s. Among Cas9s even, there is considerable diversity, and I performed phylogenetic analysis to place Ignobicas9 into its subtype. You can see that it resides down here in the tree. In this tree of representative Cas9s, the shading represents the 3 different subtypes, 2A, 2B, or 2C, that they are. I was very interested in knowing Ignobicas9's subtype because there are different patterns with respect to the sequence and structure of its guide RNA. This proved very handy in deducing what the guide RNA would be, but also provided some helpful leads in coming up with this PAM sequence, since I was starting from just the genome fragment that we had. Before moving on, I'd like to point out a few important Cas9s in this tree. The canonical SpyCas9 that we have discussed falls into the 2A subtype. It's important to note that thus far, 2 thermostable Cas9s have been reported, and they are also in the 2C subtype. They're from the Bacilli class, and they are active at temperatures up to 70 degrees Celsius. Finally, in determining the PAM, it was useful to look at in vitro validated homologs from the 2C subtype. The one to which Ignobicas9 is most closely related comes from P. labamenti virans, a bacterium with an optimal growth temperature of 30 degrees Celsius. Its PAM will prove handy in just a few slides. Knowing that Ignobicas9 was a 2C Cas9 was incredibly helpful because I now knew which features its guide RNA needed to have. With that in mind, I searched near the CRISPR array for matches with a repeat sequence and looked for matches flanked by potential promoters and terminators. I combined candidate tracer RNA and cRNA sequences using RNA folding models to see if the required features emerged and if they persisted at elevated temperatures. And that's what I showed here on this slide. Having expressed and purified the Cas9 and transcribed the RNA that I had figured out, I needed to figure out Ignavi-Cas9's PAM sequence, the last component required for a working Cas9 system. I used the known 2C PAMs as my starting point, and I tested whether Ignavi-Cas9 could cleave DNA templates with PAMs from other 2C Cas9s. I found that Ignavi-Cas9 did indeed cleave the DNA containing the PAM from P11 lentivirons. And that's what I show in the electropherogram at left. It shows the distribution of sizes of DNA present after a cleavage reaction. Longer DNA fragments take longer to run, and thus they appear more to the right of this plot. In this electropherogram, I show 3 curves. The first, in blue, is from the reaction with a template containing the PAM from P11 lentivirons. The second, in orange, is from a reaction in which the guide RNA had a match to a scrambled version of the DNA target. The third one, in green, is from just the guide RNA by itself, so I have an idea of where it appeared. You can see that the blue curve has a small but distinct peak that I was very excited to see. It was the first time that I had ever tried expressing and purifying the protein, and it seemed to be doing what we wanted it to. I felt like the phylogenetic analysis I showed earlier was very helpful, since the PAM came from the most closely related validated homolog. Even though type 2C Cas9s are known to not be as efficient as other Cas9s, I thought that I could certainly get Ignavi-Cas9 to perform better by refining the PAM sequence. To do so, I looked to bioinformatic analysis of environmental sequencing data. You can see now that the informed PAM I came up with did indeed improve the performance of Ignavi-Cas9. What I did to come up with this new sequence was to use Ignavi-Cas9's spacer array. I looked for those spacers in the bulk sequencing data from the environmental sample that had identified Ignavi-Cas9, and I isolated sequences flanking the spacers for what might be the PAM. I used edit distances to filter out those similar to the repeat sequence to make sure I was getting hits from possible viruses or bacteria that could have infected the bacterium Ignavi-Cas9 came from. From this work, I came up with a sequence logo that prompted me to adjust the PAM at the underlined 2 positions. This certainly was a nice improvement, but I wanted to settle the question of what the PAM was once and for all. I performed a substitution assay starting from the improved PAM sequence I showed you on the previous slide. I tested Ignavi-Cas9's ability to cleave DNA flanked by PAMs with each possible substitution at each of the first 10 positions downstream of the DNA target. So as shown for the first position, I tested the 3 PAMs that had a C, G, or T instead of the A nucleotide. For the second position, I tested the 3 PAMs other than those with a G, and so on. To concisely represent the data, I did the following. For a template containing a given PAM, I looked at the area under the curve of the larger peak generated from Ignavi-Cas9 cleavage. I compared its size to the area under the curve corresponding to the uncut template to come up with a cut to uncut ratio. I did this so that I could concisely represent the data from the electropherograms. What we expected was that substitutions giving negligible cut to uncut ratios would tell me that the position in question was important and not flexible because cutting was no longer possible. So for example, at the first position, all 3 substitutions, C, G, or T, seemed to be reasonably tolerated, indicating that the PAM could contain all 4 nucleotides at that position, which is what we represent by the N nucleic acid code. However, at position 2, the 3 substitutions have different effects. The A substitution is permissible, whereas the C and T substitutions dramatically reduce Ignavi-Cas9's activity. Since A and G are recognized at the position, the PAM at the second position is represented by the R nucleic acid code. I used a similar approach for the rest of the PAM, and I don't show positions 7 through 10 because we didn't find that they contributed to the PAM. Interestingly, I found that the 5th and 6th positions were an A and T nucleotide, as in P11 lentivirons. Taken together, I had the PAM completing the puzzle and giving me a functional Cas9 system. Finally, I could address the question I was burning to answer. At how high of temperatures could Cas9 cleave DNA? After all, Cas9 has an upper temperature limit of 44 degrees Celsius, and Cas9 and Cas9 work at up to 70 Celsius. The previous experiments that I showed you were actually performed at 52 Celsius. So the question remained, what was the upper limit of Cas9's activity? To get a sense of how well Cas9 could work at different temperatures, I elected to measure its efficiency, and I did that by taking the ratio of the area under the larger cut peak from the experimental reaction, which is shown in light blue, to the area under the curve from an uncut peak from a scrambled control reaction. With 3 replicates at 7 temperatures each, I found that Cas9 is able to cleave DNA from moderate temperatures all the way to 100 Celsius with reasonable efficiency. I extended this approach to conduct kinetic characterization of Cas9 at the temperatures previously presented. I found that Cas9's cleavage rate constant, which is shown in gold, is greatest at 55 degrees Celsius of those temperatures tested. And while it may not cut as quickly as Cas9 shown in gray, at the more moderate temperatures at which both are active, Cas9's wide temperature range is impressive and unique among Cas9 proteins. Motivated by the ability to cleave DNA at such elevated temperatures, I thought about applying Cas9, and amplification reactions immediately came to mind. After all, polymerase chain reactions are ubiquitous in molecular biology and genomics, but their performance can be hampered by the amplification of undesired templates like primer dimers, adapter dimers, and contaminants. And while undesired templates can sometimes be physically excised or removed using other size exclusion methods, their presence can in turn compromise the bandwidth of downstream sequencing. We considered the PCR step of the bacterial RNA-seq workflow to be an excellent opportunity to test the ability of Cas9 to target unwanted sequences, since reads from 16S rRNA tend to predominate and limit the number of reads from genes of interest. Hence, we added Cas9 to the PCR step of the RNA-seq workflow. I can add here that while a mesophilic Cas9 could be used in an additional workflow step prior to amplification to cleave undesired fragments in the library, concurrent amplification and targeted depletion offers multiple opportunities to cleave the target and prevent its exponential amplification without adding any steps, and also without the need for a cleanup step that could result in sequence loss, as with the dash method that I described. So with this in mind, I designed guide RNA to target cDNA resulting from 16S rRNA and included Cas9 complexed to the guide RNAs in the PCR step of the preparation of a library derived from E. coli RNA. As a control, I performed an identical reaction but with Cas9 instead of Cas9. I sequenced the starting library, and on this slide present the normalized coverage across the 16S rRNA region. I now overlay coverage from the control reaction, which included Cas9 and corresponding guide RNA, instead of Cas9. You can see that these curves look reasonably similar. In contrast, when Cas9 is added to an amplification reaction, targeted depletion during amplification reduces the presence of 16S rRNA and demonstrates how Cas9 can be applied. So having taken you from a sequence fragment to a validated Cas9 homolog with a demonstrated application, I'll wrap up this portion of the talk here. We discovered Cas9, which is an exciting new addition to the CRISPR-Cas9 toolbox. It expands the range at which nucleic acid cleavage is possible to 100 Celsius and demonstrated its utility in improving molecular biology workflows. In the work I described, we explored and learned from nature, and in the process, developed a technique that I anticipate will improve molecular biology and genomics workflows. In my talk today, I have demonstrated how genome editing can be used to engineer living systems to create new capabilities by recording cellular histories into their genomes. I also presented Cas9, an exciting Cas9 that underscores the power of learning from nature to enable and develop improved workflows and new applications. Looking forward, I am excited to leverage my skills in creating new technologies to tackle the complex challenge of cancer. As I shared at the beginning of my talk, the intricacies and open questions of cellular differentiation and evolution motivated me to pursue my PhD in the first place. Cancer represents one of the most far-reaching and devastating ways in which these phenomena manifest themselves. In the fall, I will join MD Anderson as an Institute research investigator in computational biology and join their efforts to identify and develop new therapeutics for cancer patients with unmet needs. With that, I'll close by acknowledging the following individuals. First and foremost, my advisor, Stephen Quake, for his mentoring, support, and the opportunity to work on the projects highlighted, as well as the colleagues I've had the privilege of working with on the work presented. I'm grateful for funding during my PhD through fellowships from the Hertz Foundation, NSF, Stanford Graduate Fellowship Program, and Siebel Foundation, along with support from the BioHub. I'm happy to take your questions at this time, and thank you for your interest and attention.
What did you use for your K-nearest neighbors algorithm and Silbar code? Did you use a specific set of software?
I just implemented, I believe, like in Python, like cycleworm. Yeah.
That's what I used. It's great.
Yes. It worked well and allowed us to get to the bottom of how we were classifying data.
It's true.
Yeah.
Great talk. I look at Navi Techman large model. You find the pro-I so further stable than other Techman.
So your questions about if the homology modeling helped us understand where the thermostability comes from. Right. That, I guess, is a question perhaps better suited to a protein biochemist, but that's something we're very interested in looking at moving forward, and that's something we're exploring. It would be great to get those insights and maybe be able to engineer that functionality into other proteins of interest. Yes.
Okay.
Okay, wonderful. So with that, I would like to take a few moments to acknowledge the people who played a very big role in my PhD. So it has been my utmost privilege to complete my PhD in bioengineering here at Stanford as a student of Steve's.
Is that the same chart?
And it is. I didn't do the same outfit, but at any rate, Steve is a pioneering leader in biotechnology, and his bold vision has led to impactful advances across so many fields. From him, I'm fortunate to have learned how to apply engineering principles to biological phenomena and to develop meaningful solutions to challenging and impactful problems. Thanks for being my advisor, Steve. I'm grateful for the guidance of my wonderful thesis committee, Professors Michael Bassick, Casey Kwong, Matt Porteous, and Stanley Chi. Their comments and advice along the way have been incredibly helpful, and I would like to thank Mike and Stanley for serving as members of my reading committee as well. I thank my Quake Lab colleagues, past and present, for a memorable and enriching experience. In particular, thank you to Norma Knapp for her timely advice and sequencing expertise, and to Brian Yu, Stephanie Zimmerman, actually from the Kim Lab, and to Stephanie Narville for being wonderful team members to work with closely. I'm also especially thankful to Whitney Combs, Bob Jones, and Sheila Cresta for making the time in the lab a productive and rewarding one. Thanks to my friends, the past few years were special. I felt at home in the Bay Area thanks to friends new and old, and so many of you are here today. I'm so grateful for that. I have lots of happy memories to look back on, and the pictures here are just a handful of them. Most of all, I'm indebted to my family. Eileen and Gregory graciously welcomed me into their family, and I'm fortunate to have had such encouraging and proud in-laws who even traveled to be here today. Thank you both. Oh, this is so bad. Someone who completely surprised me by being here today is Constantine, who may actually be wearing the same shirt in this photo as well. He is my closest friend and my younger brother, and he originally told me he could not be here today because it would be the first week of his PhD program, But thankfully, he misread the academic calendar and could be here after all. I trust that he'll learn how to read academic calendars at MIT. Maybe it's on the syllabus. But I'll stop embarrassing him after saying that I look up to him, not because he's been taller than me for a decade now. I'm kind of mad about it. But because his talent, drive, and kindness have always inspired me to merit his admiration. My parents, Eleni and Vasilios, made everything possible for me. I'm forever grateful to them for their example, their perspective, and their unconditional support. These photos from their PhD years are very special to me because I brought some of my mom's chemical engineering textbooks with me to Stanford. And on a lighter note, the model of computer that my dad used during his PhD is actually on display in the Computer History Museum. I may actually have that sweater. And finally, I am grateful to have shared this journey with my husband, Calvin. As a fellow PhD student, he has walked every step of the way with me on our journey to earn our doctorates. From applying to programs together when we were in college to walking together to lab every day. His intellect, his sincerity, and his optimism bring out the best in me, and I'm excited about what's next in our journey together. I'd like to conclude by thanking you for being here today. It's been such a pleasure to share my work with you, and this thankfully concludes the public portion of my event.